Hi, I'm Ralph Preston. Every Tuesday we have these Stroke Buddies Recovery and Science Zoom meetings where we try and bring you some useful information for your recovery. And today, back for the 9th time, and we can't thank him enough, is Dr. Bruce Hossler. Today he's going to present on the structure of the nervous system. And for anybody who would like to see his other 8 presentations, I'm going to drop the link to a playlist. He has his own playlist on my YouTube channel, and I'll put that in the description underneath the video. And so I'm looking forward to this. I always like to learn more about the brain. I think it's real important for us as stroke survivors to know all we can about the brain and how it works. I think it helps with our recovery. So I'll get quiet now and let Dr. Hossler go ahead with his presentation. Thank you, Bruce.
Well, thank you so much, Ralph. Okay, I'll share my screen here. It'll take me a minute here, but we'll get it going. And here we go. There, the structure of the nervous system. And that's right, I'm Bruce Hossler, retired professor of Psychology and Neuroscience from Lawrence University in Appleton, Wisconsin. Here we'll begin with a picture of some neurons in the brain. The human brain has 100 million neurons. And I used to tell my students that I expected to learn half of them by name. Now, normally, you cannot see individual neurons. The neurons in this slide have been stained with what's called a Golgi stain. That's why you can see them. Normally, you can't differentiate neurons from the various other cells in the nervous system. There are various supporting cells. There are astrocytes and oligodendrocytes and microglia, as well as the 100 million neurons. And the 100 million neurons differ in shape, size, and function. But Camillo Golgi developed a method for staining neurons with silver nitrate, and for that, he won the Nobel Prize in Medicine or Physiology in 1906. I was in graduate school at the time, and we were really excited to learn that we can now see individual neurons in the brain. Here's a picture of a prototypical neuron. Okay, this is just a drawing, but it does show the components of a neuron. At the top, you can see the cell body or soma that has the nucleus of the cell. There are various dendrites extending from that. Dendrites serve to receive information from other neurons. And there is a axon that's extending down. So the axon goes down and ends in terminal buttons. It's from the terminal buttons that a message is sent from one neuron to another. You'll also notice that there is a myelin sheath around the axon. I'll say more about that in a minute. The function of the myelin sheath is to speed up the rate at which a message goes down the axon from the cell body down to the axon terminals. Multiple sclerosis is a disease that selectively attacks the myelin in the brain. It doesn't damage the neurons, it goes after the myelin, disrupting communication. Okay, neurons have 3 parts, which I've already mentioned and shown you. Soma or cell body, the dendrites that receive information from other neurons, and the axon, which sends information to other neurons. And most axons are wrapped with a myelin sheath, which is a fatty insulating coating. Now, the myelin is composed of lipids and a high concentration of cholesterol. Cholesterol is white, so groups of myelinated axons are referred to as white matter. You've probably heard of gray matter and white matter in the brain. The white matter is myelinated axons. The gray matter is cell bodies. And that'll be clear in just a minute. There's a prototypical neuron. And this shows the nervous system. The central nervous system is the brain and spinal cord, which are in gold here. All of the axon pathways, the nerves going in and coming out of the central nervous system, constitute the peripheral nervous system. There's a human brain. Looking to the left. So that is a left hemisphere. Now, the human brain is normally covered with 3 layers of tissue called the meninges. The outermost layer is called the dura mater. It's tough. The consistency is sort of like that of a, well, you may have seen at Toys R Us or something, a large ball, a rubber ball that's got a rubber band on one side. You can hold it in your hand and bounce it back and forth by tapping on it. The tap will stretch the rubber band and it'll come back. So the dura mater has a consistency sort of like the rubber on the outside of that ball. Inside the dura mater is the arachnoid, which has a space beneath it called the subarachnoid space filled with cerebrospinal fluid. And the inner layer is the pia mater. It's a very thin layer on the top of the entire brain. It's sort of like a thin layer of saran wrap surrounding the brain. Now, this shows the layers of the brain. At the top, you have the hard dura mater. Beneath that is the arachnoid membrane. Then there's the subarachnoid space and the pia mater. Now, you may have heard of a subdural hematoma. Okay, that's something that's below the dura. Now, the next slide will show you. Okay, if you look at the top right, number 2 there, you see a subdural hematoma. It's below the dura mater in the brain. I hope you can see that. If you look at number 4 there, that's an intracerebral hemorrhage. That's a sort of stroke both Ralph and I had, where a blood vessel bursts inside the cerebrum. Can you all see that there?
Mm-hmm.
Okay. Now, the brain also has some spaces inside that are filled with cerebrospinal fluid. These are called ventricles. And there are 2 what are called lateral ventricles. Those are the larger ones at the top. And then you have the 3rd and 4th ventricle that go, well, the 3rd ventricle is sort of a pancake-shaped thing in the middle of the brain. And the 4th ventricle is in the brain stem. But these are filled with cerebrospinal fluid. And in some diseases, like Alzheimer's disease or alcoholism, you have an increase in the size of these ventricles. And since 2 things cannot occupy the same space at the same time, if the ventricles get bigger, that means the brain gets smaller because more space is taken up with cerebrospinal fluid rather than brain tissue.
What's their purpose, or are you about to cover that?
I wasn't going to cover too much. You need cerebrospinal fluid. It flows over your brain, and in the 3rd and 4th stages of sleep, so-called slow wave sleep, your brain basically washes itself with cerebrospinal fluid. And that clears away things that contribute to Alzheimer's disease. So beta amyloid and tau protein are literally washed away in stages 3 and 4 of sleep because of the cerebrospinal fluid going throughout the brain. And it's produced inside these ventricles, but then gets inside the rest of the brain during stages 3 and 4 of sleep, because spaces in between the supporting cells in the brain, the astrocytes, um, get larger, allowing the cerebrospinal fluid to literally bathe or wash the brain and carry away waste products.
So it's like the windshield washer fluid reservoir on your car.
So that's a good analogy. Never thought about that, but yes, that's correct. Now, this is a coronal section through the brain. Coronal means going from top to bottom, sort of straight down. And if you look at on the left shows you where the section is taken from, and the right side, if you look carefully, you can see the difference between the gray matter and the white matter. The gray matter is gray, it's labeled in the upper right-hand corner. The white matter, shown on the left side at the top. Again, the white matter is those myelinated axons. The gray matter consisting of the cell bodies. Now you can also see there are bumps on the brain, and a bump in the brain is called a gyrus. That's illustrated in the upper right-hand corner. A groove in the brain is called a sulcus. Now, why is our brain so convoluted? Well, that was to allow for an increase in the size of the surface of the brain. Evolution had basically two choices. One, the brain could be smooth and just get bigger and bigger and bigger. Well, you need a bigger and bigger and bigger skull to house that. Or the brain could be folded in on itself. And you could increase the size of the cerebral cortex, that is, these outer layers, by folding it in on itself and give a larger uh, area. And that's what evolution chose. So again, in this slide, you can see white matter and gray matter. Also, the top of the brain is called the dorsal portion. That's illustrated at the top. And the underside of the brain is called the ventral portion. So dorsal versus ventral, and gray matter versus white matter. A bump is a gyrus. A groove is called a sulcus. That's labeled on the left-hand side, although a very large groove is called a fissure. Now, again, the picture of the brain with all the various gyri and sulci is on the right. On the left-hand side, you can see the 4 lobes of the brain. In that picture, they're color-coded. Obviously, the brain normally is not color-coded, but the frontal lobe there is shown in blue. That controls movement, among other things. Behind that is the parietal lobe. That's where your body senses occur. That's where you feel touch, warmth, cold, and pain in the parietal lobe. Down below the parietal lobe and the frontal lobe is the temporal lobe. That's the lobe that's involved in hearing. Then the occipital lobe, shown in yellow, is where you see. Information has to reach the occipital lobe for you to consciously see it. Now, since the occipital lobe is at the back of the brain, why is it that when a boxer gets hit on the chin, he may see stars? How can being hit on the chin cause you to see something? Well, the brain is normally floating around in a sea of cerebrospinal fluid. When the box is hit on the chin, that thrusts the brain backwards, and the occipital lobe hits the inside of the skull. And doing that causes some of the neurons in the occipital lobe to become active, and the person sees stars. Okay? Now, each hemisphere, as we saw in the preceding figures, has four lobes: the frontal, parietal, temporal, and occipital lobes. I mentioned that the frontal lobe is involved in movement control. The parietal, temporal, and occipital lobes each have a primary cortex that provides conscious awareness of sensory experience as well as the processing of that information. So your parietal lobe has a primary somatosensory cortex that is where you feel things. The temporal lobe has a primary auditory cortex where you hear things. And the primary occipital lobe is where you see things. If anyone has any questions at any time, just say something and I'll try to answer them. Okay, here again, we have color-coded lobes of the brain. Now, this brain again is looking to the left. The frontal lobe is in blue. The very back of the posterior part of the frontal lobe in dark blue is the primary motor cortex. That's the part that contains the neurons that control body movements. So you can't move your body until a neuron in this primary motor cortex in dark blue sends information from there all the way down to the spinal cord, which relayed out to the muscle, and the muscle moves. Your whole body surface is mapped out on the cerebral cortex there. Behind that, again, is the parietal lobe. That's in orange here. The front or the anterior part of the parietal lobe, again, has a map of your body, but this is a map that allows you to feel things. If you shake hands with someone, you don't feel their hand until the information comes from your hand into your spinal cord, gets relayed up to your primary somatosensory cortex in your left hemisphere, and activates neurons there, and that's when you feel their hand. In green, down below, the temporal lobe, there's a dark green portion. That's your primary auditory cortex. That's where you hear something. And again, the left temporal lobe allows you to hear things that come in from your right ear. Your right temporal lobe allows you to hear things that come in from your left ear. And then at the back of the brain, you have the occipital lobe, the primary visual cortex at the very back. Any questions?
We had one about central nerve pain. Do you know anything about how central nerve pain happens in the brain from stroke?
Central nerve pain, which was initially called thalamic pain, but now is generally called post-stroke pain. It was
Okay.
initially called thalamic pain because it was thought to occur when your thalamus. Ralph and I both have thalamic damage from our strokes. But it mainly occurred following thalamic pain and or following thalamic damage. Luckily, I never had that. I don't know if Ralph did.
No, not really. No real pain.
But post-stroke pain generally cannot be treated with medication. The best treatment is mindfulness meditation. That seems to be what can help relieve the pain. I don't know that much more about it than.
Well, we did a talk with Thomas Haynes, who in a neuro PT in England, and on central nerve pain, and he said basically the same thing. They try medications, but they don't really work. There's not any kind of go-to medication for it, and he also mentioned mindfulness. It's a tough one because some people have it kind of bad and for long periods of time. Judy asked that question. Judy, do you have central nerve pain, do you think?
Yes, I do, quite seriously.
Well, that's why you'd probably like to know what the answer is.
Yes, I know it's a disruption of the messaging that's producing the pain. I was hopeful that Dr. Hetzler might have some ideas for me. I'm trying medication that is not working. I'm about ready to change. And the sad thing is that it's really affecting my recovery because on days that it's very serious, I have trouble walking. So it's pretty serious for me.
All the people I've ever discussed it with report the same types of things. Medication doesn't do much, and when it gets bad, it kind of shuts them down or semi shuts them down.
It's
Yeah,
true.
I wish I could be more helpful, but again, I'm very thankful I don't have that because again, my thalamus was damaged, and it used to be called thalamic pain, but
Does it come from the thalamus, or did they revise?
well, that was the area from which it was most reported.
Ah,
Yes.
And I was, I asked my physiatrist at the time if I had developed that, and he said probably not, because in his experience, it usually came from the most serious stroke. And I thought mine was serious, but I guess it could be more serious. And he said it tended to occur in older patients. Now, I was 62 at the time, uh, but he tended to think, I know, people in their 80s or so. I mean, again, that's just what he told me. And the thalamus has about 8 separate nuclei, so it's unclear exactly which nucleus has to be damaged to produce that in the thalamus, but it can come from damage to other parts of the brain. And the reason pain medication doesn't really help is that pain medication is designed to block pain impulses coming up the traditional pain pathways. I don't talk about those pathways in the brain, but there are pathways that specifically bring in pain information from the surface of the body. They have pathways, and pain medications block either the pain receptors that start them or block synapses, that is, communication signals in the pain pathway. But the human brain doesn't have pain receptors in it. So the post-stroke pain does not come from activation of pain receptors. So it's very difficult to treat.
So once you get it, then what? You're stuck with it for life?
Well, again, I'm not an expert, I'm not a physician, but uh, that's sort of the impression I got. But, it depends on how well your brain recovers from the stroke. And again, the only thing that I've read about helping is mindfulness meditation. And for that, a psychologist would probably be better able to help you than a physician. There, there are psychologists that deal with mindfulness meditation.
7:30.
So that would, if I had it, that's what I would try to do, is find a psychologist that can help me with mindfulness meditation.
Is there any kind of exercises you can do if you have it?
They're mental exercises. They're not physical. It's not, again, I'm not a physician. I'm not a clinical psychologist, but it, there, you have to basically distract your brain
Mhm.
and think about it in different ways. A clinical psychologist that deals with mindfulness meditation will be your best bet at help. And I'm not a clinical psychologist, but that would be your best bet, I think.
Thank you.
You're welcome.
One more thing, I'll let you move on. Judy, I just looked it up. I'm talking with Dr. Winston, who's the one who's doing cryotherapy on for spasticity, to talk to him about it. I looked up whether cryotherapy has been involved with central nerve pain, and it has. It's been explored as a treatment. So I'll send this to you, and
Great.
I'll try and remember to talk to, I'll definitely remember to talk to Dr. Winston about it, assuming I can get him to come on. He's kind of a big deal and pretty busy, but his assistant wrote to me after somebody I coach was about becoming a patient and asked if they would be willing to appear, which is a great
Oh.
reason to coach people for free. It sometimes comes back to you. Often comes back, always comes back to you. Anyway, sorry about that, Dr. Hetzler. Go ahead.
No problem. I'm happy if you or I can provide some sort of help. And that's what this whole program is about, helping people.
Right, and this is a big problem for a lot of people.
Yes. Okay, continuing with the organization of the nervous system, a lot of remember axons, that's the extension from a neuron cell body that sends information. Many axon pathways beginning or ending in the cerebral cortex are crossed. That is, areas in the right cerebral cortex receive sensory information and control body movements on the left side of the body, and vice versa. That is, cells in the left side of the brain receive information from the right side of the body and control the right side of the body. This is called contralateral control. Now, that's interestingly true for all of the senses except taste and smell. So if you smell something with your right nostril, it goes to the right side of the brain. If you taste something on the left side of your tongue, it goes to the left side of the brain. Taste and smell are our oldest senses. All the others are newer. So the newer ones, hearing, vision, touch, pain, those are contralateral. They cross. Now, for visual information, it's the same idea. If you look straight ahead, everything to the left of the center of your visual field is your left visual field. Everything to the right of the center is your right visual field. So information from your right visual field goes to the left side of the brain. Information from your left visual field goes to the right side of the brain. Now, this shows the layers of the cerebral cortex. Your cerebral cortex, that's the outermost layer, outermost portion of the brain, has got 6 layers. Now, there are three panels here. The middle one just shows sort of average visual cortex. On the left side, the left frame or left image shows visual cortex that's receiving sensory information. This would be like in your primary visual cortex or something. And information from coming in is in layer 4. That's the blue layer. And as you can see in the left image, the blue layer is larger because it's getting all this incoming sensory information. Now, the fifth layer, that's the pink layer, the origin of the outgoing information from the cerebral cortex. So the right image is from the primary motor cortex, which controls body movements. And you can see layer 5, the pink one, is very, very large because it's sending all this information out to your body surface to move it around. Okay? Now this shows,
Wait. If it's going to the right with all the information, get it all on the information go to the right. It's like the blue in the first one and the second one.
well,
Left.
these are images from different parts of the brain. So
Yeah, but you said left and right. It's like,
well, but this is just the left and right picture here. See, there are three pictures showing the way the cerebral cortex is organized in different parts of the brain. So the vertical column on the left-hand side is taken from the primary sensory cortex. It could be like the somatosensory cortex receiving information from the body surface. And layer 4, the one in blue, is bigger because it's getting all this incoming sensory information. The image on the right, and these are just drawings. Uh, these were drawn by an artist. This was taken from a textbook that I used when I took a neuroanatomy course at Northwestern Medical School. And these were all drawn by an artist. These are not actual brain slices. So the one on the right shows what goes on in the primary motor cortex, and layer 5, the pink layer, is bigger because you've got a whole bunch of cells there that are sending information down to the spinal cord, out to the muscles to move the body around. Now, this shows how neurons in the brain develop over years. So when you're born, the left-hand image there, you don't have many neurons in your cerebral cortex. By the time you're 6 years old, you've got a whole bunch of them. In fact, you've got too many. And they have to be weeded out. So a lot of them die off then. By the time you're 14, that's the image on the right, that's your normal amount of neurons. Now, what determines which neurons survive at age 14 and which determine ones that die by age 14? Well, it depends on the connections that they have. If neurons in your primary visual cortex and the occipital lobe at the back of your brain get information from your eyes, then they stay alive. If they haven't gotten any information from your eyes, then they die. So if you, and this has been done, if the eyelids of a kitten are both sewn shut, so they get no visual information coming into them, in the first let's say 6 months of life, and then you take the sutures out, they will be blind because none of the neurons in their occipital lobe got information. And so they died out.
So a person that's blind that's it for them then, right?
That's correct. If they're blind from birth, they're blind forever because the neurons in their occipital lobe have not received visual information, and not having that, they're all gone.
So does a neuron ever regenerate and like hanging around? It's like later on, couple years later, that regenerate again?
Well, the brain does make new neurons, but they only go where they're needed. And if they're not getting visual information, they're not going to go there.
It would tend to make you think that blind people would be less intelligent, or me. I'm sure I know blind people, so and they're not. So obviously, it doesn't.
They're not less intelligent. It's just the occipital lobe neurons. But remember, you got cerebral cortex
Right.
all over your brain. So, I mean, look at Helen Keller. She was blind. She was brilliant.
Right.
So it's just the occipital lobe that doesn't do that. Now, this shows what goes on in the brain over years. And the main thing here, if you look 3/4 of the way down, you see in like blue myelination. A portion of the brain matures when the myelin gets added to the axon. And different parts of the brain are myelinated at different times. Now, we're going to return to this notion later because it's the front of the frontal lobe, so-called prefrontal cortex, that's the last part of the brain to myelinate. And that means it's the last part to mature. And that generally happens in your late 20s. We're going to return to this notion a little later on. But the front of your frontal lobe, which is where your personality resides, doesn't become fully mature, meaning myelinated, till your late 20s. So it's not really till then that you're good at making good decisions and your personality is fully formed. Again, we're going to return to that notion. Now, this shows the visual system. The left-hand portion, you're looking down from above, and you see the two eyes there and the visual field for each eye out in front. And the information goes from the visual field to the retina in the eye, then it goes down the optic nerve to the optic chiasm, which would be labeled B there, the optic tract, to the thalamus. That's not labeled this one, but the thalamus is where Ralph and I had our strokes. If it had affected one of the nuclei in the thalamus, the lateral geniculate nucleus, we would have lost vision in part of our visual field.
Thank you, whoever.
Yeah. And then it goes over the optic radiation to the visual cortex. And we'll talk about the corpus callosum. This is a band of axons that interconnects the two hemispheres. It allows the two hemispheres to work together in unison. If the corpus callosum were severed, you would have what's called a split brain, where each half of the brain could think separately and act separately.
Wow. That must be a mess.
Yes. Okay, this is a coronal view again, going up to down, and it shows the corpus callosum severed at the top there. Sometimes this happens as in a war injury, or surgical treatment for some forms of epilepsy, or to get extra credit in an introductory psychology class, whatever. And here again, we're looking down on top of the brain to the left. It says left brain. It's really left hemisphere, right brain, right hemisphere. The language centers are in the left hemisphere. Only the left hemisphere of the brain can talk. The right hemisphere is mute. It cannot talk. It has a very basic understanding of language, but it cannot talk. Only the left side of the brain can talk
In any language?
in any language. Your linguistic abilities are in your left hemisphere. Your ability to talk and understand language are in the left hemisphere. The right hemisphere has got a very basic, rudimentary understanding of language, but it is mute. It cannot talk in any language. This shows the left hemisphere of the brain, and it shows the important language centers. You could see at the front there, in sort of orange, it's labeled Broca's area. That's in the frontal lobe. That's the part of the brain that controls speech. If you have a stroke in Broca's area, you get what's called Broca's aphasia, in which you cannot talk or you talk very poorly. Randy Travis, the singer, has Broca's aphasia because of a stroke.
Pretty bad, too.
Yes, quite bad. Now, you see at the top of the temporal lobe in the left hemisphere an area labeled Wernicke's area. That's the part of the brain that controls your understanding of speech. So if Wernicke's area is damaged, you can still talk, but what you say is meaningless. Uh, your language has the normal rhythm, normal cadence. You can talk endlessly, but it has no meaning, much like the speech of a politician.
I was going to say something like that. We all know people who have affected Wernicke's area without a stroke.
Yeah. Now, there's a band of axons, it's labeled at the top, called the arcuate fasciculus, that interconnects Broca's area in the frontal lobe with Wernicke's area in the temporal lobe. It's through the arcuate fasciculus that you talk to yourself silently.
Cool.
So you talk to yourself through the arcuate fasciculus. If the arcuate fasciculus is damaged, you have a problem called conduction aphasia, in which you have problems repeating things. You can't simply repeat something.
You can't repeat it.
You cannot repeat it.
So
For
yeah,
example, if you're asked to repeat the word quarter, you might say 25 cents. You're more likely to come up with a definition. There's some things that are easier to repeat, like ball. You could say ball. But more complicated things, you're more likely to come up with a definition.
Hmm.
Then at the back of the temporal lobe, there's an area called the angular gyrus. That's an interesting area because that converts visual to auditory information and auditory to visual information. For example, if you want to say something, you have to, let me make it easier. If you want to read something and understand what you're reading, you are looking at a visual image and you have to convert it to an auditory form to understand it. And that conversion goes through the angular gyrus. If you want to, let's see, that's if you're reading. If you want to write something, if you want to write out a sentence, you have to convert something from an auditory form to a visual form. And that also occurs in the angular gyrus. So if the angular gyrus is damaged by a stroke, you have what's called alexia with agraphia. You can't read and you can't write because you can't make these conversions from auditory to visual and visual to auditory. Okay?
Mm-hmm.
So really, you become like a sitting dummy, then you cannot say anything.
No, you can talk, you can speak spontaneously. You just can't read something because you can't convert it from a visual to an auditory form. It has to be in an auditory form for you to say it.
So listening to a recording every day is not going to do you any good then.
Well, not if you have, again, alexia with agraphia. No.
But
And by the way, if you're talking about listening to something, remember we talked about Broca's area at the front of the brain? And damage to that makes it difficult or impossible to talk. Well, recent research conducted in Helsinki, Finland, shows that the best treatment for Broca's aphasia is listening to vocal music, not instrumental music and not books on tape. Listening to vocal music is the best way to regain the ability to talk. It's unclear exactly how that works, but it works.
Is there very much success?
Go ahead.
What?
Is there very much success in doing that?
There's research showing it, and this was a study conducted several years ago in people who had Broca's aphasia. They were divided into three groups. One group listened to vocal music for 2 hours a day for 2 months. Second group listened to instrumental music for 2 hours a day for 2 months. Third group listened to books on tape for 2 hours a day for 2 months. They got a month off, and then they were tested on their ability to talk. And the group that listened to vocal music did the best. Also, they looked at fMRI, functional magnetic resonance imaging, and they had stronger axon pathways in the frontal lobe in that group. So that's, it seems, the new best way to regain speech if you've got Broca's aphasia. Now, going back to the split brain, remember that's when the corpus callosum was damaged. Okay, let's say you have someone with a split brain, a split corpus callosum, they're looking straight ahead. If you present a series of light flashes, going across their visual field from left to right. You have a total of 8 light flashes, 4 in the left visual field, 4 in the right visual field. If you ask the person how many lights did they see, what will they say?
Probably 4.
They will say 4. And which 4 are they referring to?
Hard to say.
No,
The
because
left.
no, they'd be talking about the 4 in their right visual field, because those 4 in the right visual field goes to their left hemisphere.
Yeah.
Okay?
Yep.
Now let's say you flash 4 lights only in their left visual field. If you ask the person how many they saw, what will they say?
Probably nothing.
They will say none. If you ask them to point to the lights, what will they do?
Got it with the left.
Probably
They would
with
point
their
to
right
them with which hand?
The
Right.
right.
The left hand.
left hand.
Remember, these are 4 lights in their left visual field, which goes to the right hemisphere. The right hemisphere has a basic understanding of speech and controls the left hand. Understand how this is?
I forgot about the visual fields crossing.
There is a reported case of one man with a severed corpus callosum that was trying to hit his wife with one hand and restrain that hand with the other hand. So did he really want to hurt her? Yes and no. And that's a true story.
Sounds like a song.
Now, the right hemisphere is not stupid, it just can't talk. And this shows a sort of testing they can do of the right hemisphere. A person seated in a chair, the person has a split corpus callosum, they are looking ahead at a screen, something is flashed in the left, a portion of that, picture of a toothbrush. They can use their left hand to reach underneath a platform where they can't see what's under it, but they can reach it with their left hand and identify it by touch and can remove the object that corresponds to the image in the left visual field. So the right hemisphere knows what it is, it just can't say it, but it can identify it by touch. Now, this shows what happens if a person with a split corpus callosum is asked to copy a sample with the right and left hands. The sample is shown in the left frames, and then shows what they can do with their left or right hand. Now it's interesting. If you look at the second row there, you've got a cube. This person was right-handed. When drawing it with the left hand, they could sort of get the three-dimensional aspect. But when tried with the right hand, they can't do it. And they wrote, with my right hand. So the left hemisphere doesn't get that three-dimensional aspect. The right hemisphere does that. Now this shows, in a very simple way, how the brain divides its work from between the hemispheres. So the left hand, left hemisphere, controls the right hand. It's also used in math, language, science, logic. The right hemisphere controls the left hand It's involved in music appreciation, dance, art, sculpture, and so on. But normally, of course, they function together because your corpus callosum is intact. Now, I mentioned this before, the prefrontal cortex, the front of the frontal lobe. It's critical for making decisions, planning, evaluating things. It's involved in personality, temperament. It's the last part of your brain to mature that is fully myelinate. Which is why you really can't make good decisions, at least until your late 20s.
Does it take longer for the myelin to develop in males instead of females? They always say that men mature intellectually later than women.
I'm not sure of the research on that. But yes, women do tend to mature faster. That is true.
Yeah, not physically, but
Right.
emotionally and decision-wise.
Yes.
A lot of women 18 to 22 or whatever complain about the guys that they meet not being as emotionally mature as they are.
And that is true.
It is a point of curiosity for me.
No problem. Okay, now we get to what was called a prefrontal or sometimes a frontal lobotomy. That's where this so-called prefrontal part of the brain is removed from the rest of the brain. Agassiz Moniz pioneered the technique in 1935. Walter Freeman, I don't know if I should say improved upon it, but made it easier to do. The initial frontal lobotomy was based on two observations. A laboratory chimpanzee named Becky stopped throwing temper tantrums in the laboratory after this was done to her. And a tumor removed in a human frontal lobe did not cause intellectual damage. So it was started. Here's a question for you. What did the alcoholic schizophrenic say to his doctor? I don't know if I'd rather have a frontal lobotomy or a bottle in front of me.
Okay. Um, they don't do them much anymore.
No. They did a lot of them in the 1940s
Right.
for four reasons. A belief that brain disturbances could cause mental illness, lack of effective alternative treatments, newness of neurosurgery is a separate field, and the development of the transorbital lobotomy. So there were a lot of them done, including one of the Kennedy sisters had a frontal lobotomy. This shows the transorbital lobotomy procedure
Yee.
where a transorbital leucotome, collectively called an ice pick, was basically pushed up there or actually hit with a little hammer. And then you could hold onto the handle and swing it back and forth like the wipers on your car and sever the frontal lobes. That made it an office visit procedure. You'd go in and see your doctor, and look what happened. It was used on patients who were emotionally unstable and violent. Generally had the effect of relieving emotions, and it became common. Duther Newton reported that more than 10,000 were performed in the UK. A lot more were performed in the United States, but I don't have the exact numbers. There were unfortunate side effects. There were changes in personality, emotional reactions, They had disinhibition, that is, a loss of inhibitions. For example, there was one case of a man who had this procedure done. He was walking with his wife to the theater. And he said, "Excuse me," turned and urinated in the street, and then just continued as if nothing had happened. You lose your inhibitions. People tend to be lethargic, apathetic, irresponsible, socially withdrawn. They couldn't plan their life and were generally unemployable. So it was pretty much useless.
Is that because executive functions up there in the front also?
Yes.
So
All
executive function would, I would assume, is what you use to plan your behavior.
right. So that part of the brain was no longer functioning. Okay, here's the spinal cord, about the size of your little finger. Goes up and down the spinal column. You can see spinal nerves emanating from both sides of the spinal cord. And there's a cross section through the spinal cord. If you look on the left side here, there's this a spinal nerve, and you have incoming information coming over the dorsal part, the back part, and you can see a dorsal root ganglion there. That's labeled there. There's a cell body there. We're going to come back to that dorsal root ganglion, so remember that. That's sensor information comes in the dorsal side of the spinal cord. That's towards your back. Motor information leaves over the ventral root of the spinal cord. That's in red on the bottom there. So when you move your muscles around, it's because those motor neurons inside your spinal cord send information out to the muscles. So motor information goes out the ventral root, sensory information comes in over the dorsal root. Again, we're going to come back to that dorsal root ganglion in a while. There you can see your spinal cord and the different spinal nerves associated with it. At the top of your spinal cord, you have the cervical nerves. You have 8 cervical nerves. They go down to the thoracic section where you have 12 nerves. Then the lumbar, 5 nerves, sacral 5, and coccygeal 1. If you add those all up, how many do you get, Ralph?
Well, you get 26.
Uh,
No, you got 20. Sorry, I forgot to add the 6 at the bottom. You had 31. And for anybody who doesn't know about your backbone,
you have 31 spinal nerves.
right, but you have 26 vertebrae, I believe.
Well, I don't know, and I don't care.
Well, you have 8 cervical, 12 thorax, and 5 lumbar vertebrae. So they correspond. I guess there's a nerve coming out of between each one.
Yes. So cervical, thoracic, lumbar, sacral, coccygeal. Now, let's talk about dermatomes. A dermatome is an area of the skin that is supplied by a single spinal nerve. Each side of the body has 30 dermatomes. Yes, Ralph told us we have 31 spinal nerves on each side, but the first cervical has no sensory component, just motor. So that's why you've got 30 dermatomes. And this shows, in the upper right, you can see the spinal cord, and you see the body surface with the dermatomes laid out. Now, these dermatomes are important in understanding shingles, something you don't want to get. Shingles is a viral disease characterized by a painful rash that's localized in a single dermatome on one side of the body. 2 to 4 days before the rash occurs, there may be a tingling or pain in that area, in that dermatome. A single spinal nerve is involved in the production of this rash, and the rash usually heals up in 2 to 4 painful weeks. It's caused by the herpes zoster virus. It's a reactivation of the herpes zoster virus. This is the virus that causes chickenpox. After the initial chickenpox infection, the virus remains in an inactive form in a spinal dorsal root ganglion. Remember I showed you that image of the spinal cord, the dorsal root ganglion? So that herpes zoster virus is living there, but it's just vacationing, not doing anything. But sometimes it becomes reactivated and it travels down the axon back to the skin surface where it causes shingles. Okay, there's that picture again. So you've got the lower left-hand side, you've got that dorsal root ganglion, and that virus is living there. When it becomes activated, it just follows down the lower left, back out to the skin surface. Now, there are risk factors for developing shingles. First, you have to be old, like me, at least 60 years old, having poor immune function, having had chickenpox before 18 months of age. But luckily, there's Shingrix, a vaccine for people age 60 or older that will prevent this. Okay, there's your brain cut down the middle, mid-sagittal cut. So let's talk about what's in that. We'll start at the brain stem. At the very top of the spinal cord, you have the brain stem. This includes the medulla and the pons. Behind that is the cerebellum. And at the top of the brainstem is the midbrain. Okay, if you look at the bottom of this image, you see the brainstem there. At the very bottom of the slide, you see the 3 parts of the brainstem. The medulla, which is just above the spinal cord, then the pons, and then the midbrain. On the back of the palms is the cerebellum, which looks sort of like a head of cauliflower stuck to the back of the brainstem. Okay, we'll start with the medulla. The medulla controls the so-called vital reflexes of the body. It regulates heart rate, digestion, respiration, that is breathing, blood pressure, coughing, and vomiting. Drug overdoses, for example, with fentanyl, the drug that seems to be in just about everything you buy on the black market now, causes depression of the breathing center in the spinal cord. So you die from fentanyl because you stop breathing. Because all opiates, of which fentanyl is one, suppress the respiratory center. And you just stop breathing. That's how it kills you.
Can you have a stroke in the medulla?
Yes, they are usually fatal. But yes, you can.
Seems like that would be either kill you or be devastating.
Yes. They would usually be fatal.
Usually be fatal. Okay.
Yeah. Now, since the medulla also controls vomiting, opiates will also often cause vomiting at the same time you stop breathing. That's how Jimi Hendrix died.
And Elvis Presley.
Overdose. He basically suffocated on his own vomit. Now, above the medulla is the pons that contains sleep centers. One of them is the locus ceruleus, and the locus ceruleus produces a neurotransmitter, norepinephrine, which is important throughout the brain. Recall behind the pons is the cerebellum. It's involved in movement and balance. It integrates sensory and motor information and controls rapid movements and hand-eye coordination. For example, if you're playing tennis and you see a tennis ball coming towards you, you have to coordinate that incoming visual information with the outgoing motor information so you swing the tennis racket at the right time so you hit the ball. And that control of movement is done by the cerebellum. If you have cerebellar damage, you tend to get ataxia, a tendency to stumble and fall, and hypotonia, a loss of muscle tone. The midbrain's got a lot of stuff in it. At the top or the dorsal portion, you have the superior colliculus and the inferior colliculus. The superior colliculus controls eye movements. It does so by controlling certain cranial nerves, which we will get to, and thereby controls eye movements. The inferior colliculus is part of the auditory system. The middle of the midbrain includes the ventral tegmental area. You have dopamine neurons there. Those dopamine neurons release dopamine, and that makes you feel good. When you feel good because you've gotten an A on an exam, or you just scored a touchdown, or whatever, your brain releases dopamine neurons that were, well, from the ventral tegmental area, they release dopamine basically in the hypothalamus, which we'll get to. The substantia nigra is also an area in this middle part of the midbrain that has dopamine neurons. And when the substantia nigra dopamine neurons have problems, that's when you develop Parkinson's disease. The reticular formation is also here. That's a collection of nuclei that control attention and arousal. The main cause of coma is damage to the reticular formation in the midbrain. Okay, here again, here you see the medulla at the bottom and the pons, the midbrain, and behind that, the cerebellum. This shows the reticular formation. It's really a central core of much of the brainstem, but the most important part is in the midbrain, which is labeled ME, and a stroke there will cause a permanent coma. This is a cross section through the midbrain, and I show you this to show you where, excuse me, the substantia nigra is. The substantia nigra has cell bodies that send their axons forward in the brain and release dopamine in a part of the brain called the basal ganglia. This is an actual slice through a human brain that's been stained, showing where the substantia nigra is. And again, when those neurons there die, you get Parkinson's disease. First described as the shaking palsy in 1817, my first year of graduate school, by James Parkinson. It's the second most common neurodegenerative disease, prevalent in 1% of the population over the age of 60. Here are two people, Muhammad Ali and Michael J. Fox, who both have Parkinson's disease. Symptoms include vibratory movements of the arms and hands that diminish when you do something. This is called a resting tremor. Basically, if you're not doing anything, you're shaking. Your joints are rigid. You have what's called bradykinesia. Your movements are slow and you're unstable. It's a progressive neurological disorder. That is, those cells in the substantia nigra naturally die off the more you age.
Is there a particular time when you start getting this?
Yes, you start getting the disease when 80% of them are dead. At birth, you've got 100%, and when you've lost 80%, that's when you start getting the symptoms of Parkinson's disease. It's a just gradual degeneration of these neurons. It occurs naturally. Most people die before they get a loss of 80% of these nerve cells. So the vast majority of people don't get Parkinson's disease. The natural cause is unknown. You can have symptoms that mimic Parkinson's disease from a stroke or encephalitis, if your brain swells, for example, or head trauma. And we now know that exposure to compounds used as pesticides and herbicides, like Roundup, can cause Parkinson's disease. When I say exposure to them, that means, for example, if you've got Roundup and you spill some on your hands, well, that can be absorbed by your skin and it gets into the bloodstream, gets to these cells in the substantia nigra and kills them off. Now, you're not going to develop Parkinson's disease immediately then, but I don't know if you can see my hand up here, but let's say at age 30, well, let's say the cells in your substantia nigra are normally dying. At age 30, you spill Roundup on your hand, then you have a rapid loss of cells there, and then the cells naturally keep dying off, and you reach that loss of 80% earlier than you would have. So if you get Roundup on yourself now, well, it may take 20 years before you lose that 80% of those neurons, and then you develop Parkinson's disease. My mother died of Parkinson's disease. We aren't sure why, but she worked at Shaw's Garden in St. Louis, and we suspect that she came in contact with compounds used as pesticides or herbicides, and that's what caused it. But you never know for sure. This shows the pathway that I'm talking about. Substantia nigra down there in the midbrain sends its axons forward to, here it's labeled caudate and putamen, that's part of the basal ganglia. And that axon pathway just naturally degenerates the older you get. Any questions? Okay, we'll move on to another part of the brain called the diencephalon. That includes the thalamus and the hypothalamus. The thalamus receives sensory and motor information and passes it on to the cerebral cortex. It's because of the thalamus that you have selective attention. You can concentrate on specific things and ignore other things. The thalamus looks like basically an egg in each hemisphere. This shows it here, shows how it projects to the cerebral cortex. A better image is shown here. Again, this is a drawing. It was taken from that same textbook that I used when I took the neuroanatomy class. You see the different nuclei in the thalamus in the lower frame, and then parts of the cerebral cortex that they project to. Hypothalamus receives information from the internal environment and coordinates information to maintain homeostasis. It regulates body temperature, salt-water balance, hunger, thirst, energy, metabolism, reproduction, and emotional responses. Hypothalamus is very important. It's right above the roof of your mouth, the hypothalamus.
Hmm.
And this shows it here. And you don't have to remember those portions. The telencephalon, that's the large part of the brain we began talking about, includes the cerebral cortex, the basal ganglia. Those are those motor structures that receive dopamine information from the substantia nigra. Remember, that's what's involved in Parkinson's disease. The limbic system, structures involved in motivated behavior and learning. The upper frame shows the basal ganglia in the brain. The lower image shows what the basal ganglia look like outside the brain. Sort of looks like a creature but that's inside your brain. The limbic system, a lot of structures, anterior thalamus, amygdala, septal area. First described by, it's pronounced papes. It looks like it would be pronounced papes, but papes is the pronunciation. A group of subcortical structures that are involved in emotional behavior, memory, and motivation. It shows some of them identified in the midsection of the brain. There are some others. You can see the amygdala down there. That's important in emotion. Hippocampus involved in memory. Septal areas in there. Removal of the amygdala. That's involved in taming and docility.
Why would they remove an amygdala?
Well, it was done experimentally
Okay.
in rhesus monkeys. And it produced a whole syndrome called the Kluver-Bucy syndrome, which the monkeys were quite mean initially, were quite docile after that. They were good eaters. They were also hypersexual. The Kluver-Bucy syndrome is sometimes seen in humans as a result of a viral infection of the brain that damages the amygdala and other structures. Removal of the septal area, in contrast, produces what's called septal rage syndrome. If this is done in rats, the rat will attack anything that moves, including the experimenter. And you get something like this in humans. Many years ago, there was someone with a high-powered rifle on a bell tower, I think at the University of Texas, and he killed a number of people. He was finally shot by police, and a post-mortem examination of his brain showed a malignant tumor in his septal area.
Charles Whitman.
Yes, oh, very good. The hippocampus, involved in the formation of new memories. Now, I'm sure you don't know who that is. This is the single most famous patient in all of neuroscience. This is patient HM. He had a severe memory impairment following an operation that was designed to help treat epileptic seizures that resulted from a bicycle accident when he was young. And it caused an inability for him to learn anything new,
Mm.
any new verbal memory. He could learn a motor skill, but had no memory of having learned it. For example, your knowledge that you can ride a bicycle is a verbal memory. Your ability to ride a bicycle is a motor memory. So you could learn new motor skills, but had no knowledge of having learned them. And if you were to meet him, he'd say hello, you could shake hands with him, you could introduce yourself, he could talk to you by name. If you left the room and came back half an hour later, he'd have no memory of ever having met you before. His brain is shown on the top, left and lower left, normal brain on the right. So the top image is the ventral side or the underside of the brain. You see in patient HM, his hippocampus was removed on both sides of the brain. Lower left, that's a coronal section that is top to bottom. Again, you see the area where the hippocampus was removed. On the right-hand side, you see a normal brain, the top ventral side, bottom side, coronal section. Now we get to the cranial nerves. There are 12 of them. Okay. Some are mixed, but some have only sensory or motor functions. Here you have an image of them. This is taken from that same textbook that I used in that neuroanatomy class at Northwestern University Medical School. And you have all 12 cranial nerves here. So you want to remember all of these for the test. For the test of one of my classes, I had all students give the name, number, and function of all 12 cranial nerves. Okay? And they are lined up in order on the ventral or underside of the brain. The first one at the front of the brain, that's the olfactory nerve. That's responsible for smell. The second is the optic nerve that brings in information from the eyes. The third is the oculomotor nerve that controls most eye movements. Fourth is the trochlear nerve that controls the superior oblique muscle that moves the eye. Fifth cranial nerve is the trigeminal nerve, called trigeminal because it has 3 divisions. 6 is the abducens that controls the lateral rectus movement, muscle that moves the eye. 7th is the facial nerve that controls facial muscles. 8 is the auditory vestibular. 9 is the glossopharyngeal. 10th is the vagus. You had someone talking about vagal stimulation a week or two ago.
Yep.
11th is the spinal accessory, controlling neck muscles. And 12th is the hypoglossal, controlling tongue movement. And here they are. They're indicated by Roman numerals. It's got olfactory, optic, oculomotor, trochlear, trigeminal, abducens, facial, auditory, vestibular, glossopharyngeal, vagus, spinal accessory, and hypoglossal. And if you were in one of my classes, you had to memorize all these. And I gave you a useful mnemonic device to help you do that. What you have to do is remember, on old Olympus' towering top, a fair-armed girl bends snowy hops. Easy to do.
Oh, yeah.
And the first letter of each word there is the first letter of each of the cranial nerves: olfactory, optic, oculomotor, trochlear, trigeminal, abducens, facial, auditory, glossopharyngeal, vagus, spinal accessory, and hypoglossal. Right, Ralph? So now you can say them all.
You can take the test.
You can take the test.
I actually learned all the old fact. I learned the first 6 or 7 working with a physical therapist.
Oh, good.
So I actually knew them. Yeah, you use 2, 3, 4, and 5 when you do the eye test.
Yes. Okay, the trigeminal is the largest cranial nerve. There are 3 major branches: ophthalmic, for your scalp, forehead, eyelid; maxillary, lower eyelid, cheeks, lip, and teeth; mandibular, both sensory and motor. All 3 branches join at the trigeminal ganglion, which contains cell bodies. It's like the spinal root ganglion, but this is for a cranial nerve. And it shows the areas innervated by the 3 portions. Got the ophthalmic, maxillary, and mandibular on the left-hand side of the figure there. Can you see them all there?
Mm-hmm.
Okay. That brings us to cold sores caused by the herpes simplex virus. The herpes simplex virus is the cause of cold sores around the mouth. The virus is transported along the axon from the site of the initial infection to the trigeminal ganglion. Remember the trigeminal ganglion? Well, I don't really have a picture of that, but it's like the spinal root ganglion. And it persists there in a latent state in the trigeminal ganglion until it's reactivated. When it's reactivated by taking corticosteroids, upper respiratory infection, stress, sunlight, it will follow the axon back to your face, and then the person gets a cold sore again. And it'll stay there until your immune system destroys it, but you'll get it again because you always have some in that trigeminal nerve ganglion. The immune system can't get to it while it's there, and when reactivated, it goes back to your face and causes a cold sore. So once you've gotten it by kissing someone else that has a cold sore, you'll have it for the rest of your life. There's no cure for it. It comes, it erupts, you got a cold sore, and then your immune system destroys it there, but it's still in the trigeminal ganglion. My father had cold sores, and that's just the way it is. Now, Bell's palsy is caused by injury to the facial nerve, which is the 7th cranial nerve. It causes paralysis of the muscles used in facial expressions. And typically one side of the face will droop. The facial nerve is located superficially and can be easily damaged. It can also be damaged by infection. Whoop. Now, it will typically spontaneously recover in a few months, but not always. I don't know if any of you have seen the Sewing with Nancy show on public television. There's a Sewing with Nancy show. She developed Bell's palsy as a young child, and it never recovered. And her mouth always drooped. At Lawrence University, Ralph Nader came to talk one time. You may not remember him. He rose to prominence when he wrote a book called Unsafe at Any Speed about a car that he thought should never be driven. He was an advocate of many things. But anyway, on the way up to Appleton, I think from Chicago, he fell asleep with his face against the window on the plane. And when he arrived in Appleton, he had Bell's palsy because the cold window damaged the facial nerve. Now that did heal a couple of months later, but that's what can happen. And speaking of what can happen, that happens to be the end of my talk for today.
Once again, for those in YouTube land, I'll put the link to Dr. Hossler's other 8 presentations in the description underneath this video. And Dr. Hossler, I can't thank you enough for, this is number 9. You've contributed a lot to our knowledge base about the brain, and I know I speak for other people. I'm appreciative, and I speak for other people when I say thank you, thank you, thank you.
You're welcome, you're welcome, you're welcome. I
Thank
mentioned
you.
that the hippocampus is involved in the formation of new memories. So what did the hippocampus say in his retirement speech? Thanks for the memories.
All right.
On that note, I'll say goodbye.
Bye
Thank
now.
you.
Thank you.
Welcome.
Do all you can with what you have, in the time you have, in the place you are. Do all you can. Do all you can.